Hello and welcome to another Science on the Edge snack size. I'm Dr. Mark Darcy, and in this snack I'm going to talk about cell death. Cell death is incredibly important in both development and in dealing with things such as cancer. So, cell death. What is it? How does it occur? And why is it relevant? Broadly speaking, cell death occurs in either a controlled, programmed way, involving a series of biochemical and molecular events, or alternatively, in a poorly controlled way, resulting in the spilling of the cellular contents into surrounding tissues. The controlled method of cell death is generally termed apoptosis, from the Greek denoting falling off, as in the leaves falling from a tree, whilst the uncontrolled form of cell death is usually referred to as necrosis, from the Greek denoting to kill. Many of the most effective currently available chemotherapeutic drugs rely to one extent or another on initiating cell death, usually by apoptosis. However, the non-specific initiation of cell death that most of these drugs induce often results in the death of non-cancerous cells, together with the death of the tumorigenic tissue. The need for the development of more targeted drugs to initiate cell death is therefore key to producing more effective clinical treatments for cancer. So let's start by talking about apoptosis. The word apoptosis was first used in a 1972 paper by Kerr, Wiley and Curie to describe a morphologically distinct type of cell death. Apoptosis is the process by which a cell ceases to grow and divide and instead enters a process that ultimately results in the controlled death of the cell without, and this is key, spillage of its contents into the surrounding environment. Apoptosis is also sometimes referred to as programmed cell death, or more colloquially, cellular suicide. The initiation of apoptosis is dependent on the activation of a series of cysteine aspartate proteases known as caspases. There are two categories of caspases, the initiator caspases and the executioner caspases. Once cell damage is detected, the initiator caspases, these include caspases A to 9, become activated. They become activated from inactive procaspases, which are kind of floating around in the cell. They go on to then activate the executioner caspases. These include caspases 3, 6, and 7. Broadly speaking, however, apoptosis can be distinguished from the other major type of cell death, necrosis, both visually under the microscope and via a number of molecular biology techniques, including flow cytometry with an exin-5 staining, and DNA fragmentation assays. In the former, the apoptotic bodies containing the contents of the dead cell can be phagocytosed by surrounding cells, although this behavior is observed primarily in cell culture. In vivo, cells such as macrophages often remove apoptotic cells before they fragment. This results in a containment of the injured tissue and as a result reduces the risk of collateral damage to surrounding cells. The process of apoptosis is highly conserved within multicellular organisms and is genetically controlled. Apoptosis can be initiated by the cell itself when it detects damage via a number of intracellular sensors, a mechanism known as the intrinsic pathway. Alternatively, it can result from the interaction between a cell of the immune system and a damaged cell, which is known as the extrinsic pathway of apoptosis. I'm going to talk about both the intrinsic and the extrinsic pathways shortly. In the human body, it's estimated that approximately 1 × 10⁹ cells undergo apoptosis per day. There's a huge turnover of cells in an adult human body. Both the intrinsic and extrinsic pathways of apoptosis work synergistically to ensure that multicellular organisms remain healthy and defective cells are removed from the body. Failure to regulate apoptosis can result in the pathologies exhibited in many diseases. For example, in degenerative diseases such as Alzheimer's, where neuronal death appears to be initiated by the activation of caspases, a key group of enzymes, remember, involved in apoptosis. Too little apoptosis, however, can result in the uncontrolled growth and division of cells that is observed in cancer. So I'll start by talking about the intrinsic pathway of apoptosis, then I'll move on to the extrinsic pathway and other forms of cell death. So the intrinsic pathway, also known as the mitochondrial pathway of apoptosis, involves a variety of stimuli that act on multiple targets within the cell. This form of apoptosis is dependent on factors released from the mitochondria and is initiated either from a positive or a negative pathway. Negative signals arise from the absence of cytokines, chemical messengers, hormones and growth factors in the immediate environment of the cell. Without these pro-survival signals, pro-apoptotic molecules within the cell, such as Puma, Noxa, and Bax, that are normally inhibited become active and initiate apoptosis. Other factors that initiate apoptosis are positive in nature and include exposure to hypoxia, so low oxygen environment, toxins, radiation, reactive oxygen species known as free radicals, viruses, and a variety of toxic agents. Although in the case of some cells, such as neutrophils, a type of phagocytotic white blood cell, hypoxia can actually promote cell survival. The initiator caspase that controls the intrinsic pathway of apoptosis is known as caspase 9, which is able to bind to adaptor protein apoptotic protease activating factor 1, a long name I know, it's abbreviated to APAF1. It binds following exposure of its caspase recruitment domain, known as a card domain. APAF1 is in a non-apoptotic cell, is usually folded in such a manner that its card domain is blocked, therefore no apoptosis. And procaspase 9, which also contains a card domain, is therefore unable to bind to it. Card domains bind to card domains. When apoptosis is induced either by positive or negative stimuli, changes are triggered in the mitochondrial membrane, the result of which is opening of the mitochondrial permeability transition pore, known as MPT. Once the MPT pore is open, pro-apoptotic proteins, including cytochrome C, protein known as SMAC or Diablo, and another protein known as HTRA2 or OMMI, are able to then leak from the mitochondria into the cytoplasm and activate apoptosis. Cytochrome C induces apoptosis by binding to the WD domain of APAF1 monomers, which results in a conformational change, a change in shape in APAF1, exposing a nucleotide binding and oligomerization domain that's able to bind to deoxy-ATP, DATP, which is kind of floating around in the cytoplasm of the cell. This binding induces an additional conformational change in APAF1, exposing both its card and oligomerization domains, thus allowing several APAF1s to assemble into a complex known as an apoptosome. The apoptosome contains in its open center several exposed card domains, which then recruit and activate several procaspase 9 proteins. Remember, caspases exist initially as procaspases, inactive proteins. Then when activated, they become caspases. So, the apoptosome contains in its open center several exposed card domains, which then recruit and activate several procaspase 9 proteins by binding to their card domains. These activated caspase 9 enzymes, which are initiator caspases, are then able to activate the executioner procaspase 3, which in the form of active caspase 3 can fully induce apoptosis. SMAC Diablo and HTRA2 OMI help initiate apoptosis by inhibiting inhibitors of apoptosis proteins, IAPs, so they inhibit the inhibitors. Although without the release of cytochrome C, inhibiting IAPs alone is insufficient to initiate apoptosis. So this all needs to play together at the same time to actually get the intrinsic pathway to fully activate and for apoptosis to be initiated. So that's the intrinsic pathway of apoptosis. I will now talk about the extrinsic pathway of apoptosis. The extrinsic pathway, also known as the death receptor pathway of apoptosis, is initiated by patrolling natural killer cells or macrophages. These are types of white blood cells when they produce death ligands, basically molecules which can induce apoptosis. These death ligands, which upon binding with death receptors on the surface of target cells, these death receptors are in the membrane of target cells, induce the extrinsic pathway via the activation of procaspase 8, which then becomes the active caspase 8. Now, death receptors are members of the TNF, tumor necrosis superfamily, and include several members, with each death receptor having a corresponding death ligand. To activate caspase 8, a death ligand must bind to a death receptor, resulting in recruitment of monomeric procaspase 8 via its death-inducing DED domain to a death-inducing signal complex disc. So these molecules come together to form this disc. This disc is located on the cytoplasmic domain of the ligand-bound death receptor. The disc also includes either an adapter protein known as fast-associated death domain, FAD, or TNFR-associated death domain, TRAD, which facilitate the interaction of procaspase 8 to the disc. The recruitment of several procaspase 8 monomers to the disc results in the dimerization and activation, with resultant caspase 8 able to then induce apoptosis via one or the other of two distinct subpathways. The particular subpathway that is induced depends on whether the cells are classed as type 1 or type 2 cells. In type 1 cells, caspase 8 directly cleaves executioner caspases and therefore directly initiates apoptosis. In type 2 cells, IAPs inhibit direct caspase 8 activation of the executioner caspases unless the IAPs are inhibited by proteins released from the mitochondria. The important role of caspase 8 in controlling the intrinsic pathway of apoptosis has been observed in caspase 8 deficient mice, which show no response to death receptor ligands. Whether or not apoptosis is triggered by the extrinsic or the intrinsic pathways, its tight regulation is essential, and failure to regulate it effectively can have dire consequences. In cancer, for example, the cell fails to initiate apoptosis due to mutations in the various mechanisms of initiation. If this occurs in tandem with the cell failing to respond to external signals that would normally provoke the extrinsic pathway or inhibit proliferation, then this causes the cell to grow and divide uncontrollably, resulting in the formation of either a benign tumour or in the development of cancer. So to kind of summarize apoptosis, we have the intrinsic pathway, we have the extrinsic pathway. The intrinsic pathway of apoptosis is when the cell realizes it's damaged and decides to enter this cycle of programmed cell death. The extrinsic pathway of apoptosis is when a patrolling natural killer cell, for example, becomes aware that the cell is damaged and tells it to go into programmed cell death. Extrinsic and extrinsic pathways, therefore, are very different, but the end result is the same. Apoptosis of the cell. The cell shrinks, compartmentalizes, and breaks apart. It's then absorbed by surrounding cells. So I will now talk about another form of cell death, known as pyroptosis. Programmed cell death with collateral damage. This differs from the kind of programmed cell death you get in apoptosis, where there is no or very little collateral damage to surrounding cells. So, classic apoptosis, both the intrinsic and extrinsic pathways, are characterized by the compartmentalization of intracellular components of the cell and removal of cellular debris, without any collateral damage occurring to neighboring healthy cells. An alternative form of apoptosis, known as pyroptosis, was identified by Boyes in 2001. Now, in this form of cell death, although following a programmed series of caspase-dependent events, it's pro-inflammatory. You actually get inflammation, which you don't get in apoptosis. This form of cell death, termed pyroptosis, has been identified in macrophages infected with Salmonella or Shigella and is not observed in caspase-1 deficient cells, which shows you the importance of this particular caspase in pyroptosis. Once activated by a pathogen, caspase-1 processes the proforms of the inflammatory cytokines IL-1 beta and IL-18 into their active forms. This results in apoptosis of the cell, but with a concurrent release of inflammatory cytokines into the environment. Since its discovery, pyroptosis has been observed in the central nervous system and the cardiovascular system, suggesting that this form of cell death is biologically significant. Another form of cell death is known as autophagy. Actually, autophagy doesn't always result in cell death, but can result in cell death. Autophagy, on the other hand, can actually protect a cell. So it sort of depends. So, autophagy, a mechanism for both protecting and killing stressed cells. So autophagy is a process where cellular compartments such as macroproteins or even whole organelles are sequestered into lysosomes for degradation. Lysosomes are basically the stomach of the cell. The lysosomes are then able to digest these substrates, the components of which can either be recycled to create new cellular structures and or organelles, alternatively can be further processed and used as a source of energy. Autophagy can be initiated by a variety of stressors, most notably by nutrient deprivation, caloric restriction, or can result from signals present during cellular differentiation and embryogenesis and on the surface of damaged organelles. So autophagy has also been shown to be involved in both the adaptive and the innate immune system, where it may degrade intracellular pathogens and deliver antigens to MHC class II holding compartments and initiate the transportation of viral nucleic acids to toll-like receptors. Although autophagy is often used to recycle cellular components, it can result in destruction of the cell, and in this way has been linked to removal of senescent cells from aged tissues and destruction of neoplastic lesions, so precancerous lesions. Failure of autophagy, as well as potentially allowing the development of cancer, has also been associated, particularly in aged organisms, with the accumulation of protein aggregates in the neurons and the development of neurodegenerative conditions, including Alzheimer's disease. To date, 3 distinct forms of autophagy have been identified. These are macroautophagy, microautophagy, and selective autophagy. In the most described form of autophagy, macroautophagy, whole regions of the cell are enclosed in double membrane vesicles referred to as autophagosomes. These autophagosomes then fuse with lysosomes to become autophagolysosomes, and the contents are then degraded by proteases present therein. In microautophagy, the cargo, organelles or regions of the cytosol, directly interact with and fuse with the lysosomes. Microautophagy is more specific than macroautophagy and can be triggered by signaling molecules present on the surface of damaged organelles, such as mitochondria or peroxisomes. This results in specific fusion of lysosomes with these organelles. Depending on which organelle is targeted, the resultant autophagic vesicle is referred to by a specific name. For example, if a mitochondria, the term used is mitophagy, or for peroxisome, the term is peroxiphagy. In selective autophagy, also known as chaperone-mediated autophagy, CMA, proteins within the cytoplasm are targeted for fusion with lysosomes by a cytosolic chaperone. This is through interaction between the chaperone and a pentapeptide present within the amino acid sequence of the substrate. Substrate proteins then bind to a lysosomal receptor, LAMP2A, and are carried into the lysosome for degradation. Notably, the phytochemical resveratrol, which is found in the skin of red grapes, amongst other things, has been found to actually stimulate autophagy and therefore protect cells. This may be part of the reason why resveratrol in various experiments has been shown to increase longevity amongst various organisms, including mice. Basically seems to preserve the cells for longer, help them recycle their contents and get rid of protein aggregates. More research, however, needs to be done with resveratrol. A lot of research has been done, more is ongoing. But it may actually turn out that resveratrol or derivative of resveratrol could be useful in extending longevity in humans. Time will tell. Now I would like to talk about necrosis. This is cell death with collateral damage. Unlike apoptosis, necrosis is an alternative, uncontrolled form of cell death that is induced by external injury, such as hypoxia or inflammation. This process often involves upregulation of various pro-inflammatory proteins and compounds, such as NF-kappa beta, resulting in the rupture of the cell membrane, causing spillage of the cell contents into surrounding areas. This results in a cascade of inflammation and tissue damage. The contents of the cell, specifically the contents of things like lysosomes, damage neighbouring cells when released. In contrast to apoptosis, necrosis is an energy-independent form of cell death, where the cell is damaged so severely by a sudden shock, for example radiation, heat, chemicals, hypoxia, etc., that it is unable to function. The cell usually responds by swelling, a process known as oncosis, as it fails to maintain homeostasis with its environment. it then usually bursts. The previous definition of necrosis as a counterpoint to apoptosis is a useful concept. However, as necrosis is usually observed as an endpoint state in cell culture by the presence of sale of fragments in the media, what is described in many cases could simply be the remains of late apoptotic cells, the apoptotic bodies of which have lost integrity. In vivo, these apoptotic bodies would be phagocytosed by patrolling white blood cells. However, in the single-cell population environment found in culture, this does not occur, which can confuse the diagnosis of the exact mechanism of cell death. A more accurate counterpoint to controlled cell death would probably be oncosis. This will be described next. So oncosis. Oncosis, cell death with collateral damage. The term oncosis is derived from the Greek word onkos, which means swelling, and is best thought of as a pre-lethal pathway that ultimately leads to cell death. Its main features are the swelling of both the cell generally and the organelles of the cell specifically, together with an increase in membrane permeability. As oncosis progresses, there is a depletion of intracellular energy stores and an eventual failure in the ionic pumps of the plasma membrane. The result of oncosis is a leakage of cellular debris into surrounding tissues and consequently damage to surrounding cells, inflammation. Oncosis can be induced by all of the methods that I previously described, or in some cases by infection of the cell by pathogens such as rotavirus. So why is cell death so important? Cell death is important because misregulation of cell death can really have profound effects on human health. As we grow from an initial single cell, a zygote, right through to an embryo, a fetus, a baby, and right through childhood to adulthood, a series of events occur whereby cells grow, divide, and die. Development is not just about the cells growing and reproducing, it's about the correct cells dying so that we can shape a fully formed and functioning human body. Dysregulation of cell death can result in all sorts of diseases. Too much cell death can result from autoimmune diseases. Too little cell death can result in cells continuing to divide until the result is a disease such as cancer, which obviously can be life-threatening. So that's cell death in brief. A lot of information that I know, and I do tend to talk fast as well. My suggestion would be to go over this particular snack size edition a few times if you want to absorb all of the facts. Okay, so that's all for now. I look forward to chatting with you guys again in the next Science on the Edge podcast or the next snack size. Have a great day. Thanks again for listening. Hope you enjoyed the episode. If you did find the episode interesting and would like to know a little bit more, there are a number of ways you can do that. First of all, you could follow me on Twitter. My handle is @Dr. Mark Darcy. That's just all lowercase, DRMARKDARCY. You can drop me an email. The address is info@scienceontheedge.com. Or you could visit the website, scienceontheedge.com. The website is still under development, but new content is appearing all the time. Also, if you would like to support Science on the Edge and keep these podcasts under production, then you can make a small donation by visiting scienceontheedge.com and clicking on the donate button. Thank you very much and hope to see you again soon.